All right, hello everybody. Welcome to this workshop from the Agricultural Genome to Phenome Initiative. I believe everyone from the waiting room is in, and we're going to try and get started right on time here. So my name is Nicole Scott. I am the project manager for the AG2PI project, and we are funded by USDA NIFA. And I would like to introduce our speaker today, Filipe Matias. He's a PhD in genetic and plant breeding with experience working with corn, tropical grasses, potato, and wheat from 8 different institutions around the world, including Brazil, Portugal, and the US. His interests are quantitative genetics, genomic selection, GWAS, phenomics, high-throughput phenotyping, remote sensing, image analysis, and R. And today he is going to be presenting on his project, Bison-Fly, which is a UAV pipeline, and in this case applied to plant breeding programs. And this is the result of an AG2PI seed grant that was awarded in our first round of funding. So Matthias, would you like to take it away?
Yeah, thank you. Thank you for the introduction. Also, thank you for inviting to be here today. I can see a couple names that I recognize here. Thank you guys for coming here. Anyway, hey, Abdallah, nice to see you. So, guys, feel free to make any question today. We just want to share and learn with each other. And I also learn a lot with questions. So make questions when you want to do. I guess maybe Nicole prefer typing the question on the chat and then she stop and make the question to me. But feel free, okay, to interact. What else? I do feel very excited talking sometimes, and I may start speaking Portuguese. So let me know if I start speaking Portuguese, okay? Just in English, I'm gonna try. Então, let's start. The main idea here is to share this pipeline that we developed with the seed grant that we approved from AG2PI last year, or 2 years ago, maybe. Max Feldman, he is here in the call. So it's nice to see you, dude. Feel free also to help me with answering questions, okay? But before we start talking about the pipeline that we in our workshop today, I want you guys to go in the link that I'm gonna share right now in the chat, which is our workshop. So I built an online machine using Binder. After you open, if you want to follow the code with me, just go in the link that I just shared and go. You come here below in this blue button called Binder, and then open a new window. Okay, so we're supposed to open a Binder, RStudio Binder, stylish, somewhere in the world where it's going to be our workshop, okay? After you do this, just come down below here and press the tutorial R. That's going to be the code that we're going to be running, right? If you want to do this in your machine, an easy way to do it is basically come here in this code part and download zip. Everything, all data that we're going to be running and information is going to be there, okay? Also, the tutorial, the scripts is going to be there as well. All right. Before we really start going all steps in the Bison-Fly pipeline, I want to start with some basic information, some introductory concepts that I guess make totally difference when you want to start applying this information plant breeding. I know that probably many of you here guys already know it, but once that this video is going to be on YouTube and many students can come to watch it later, I guess that's very helpful to understand why this is so, why we can use images, for example, in plant breeding, and how we can use these for applying selection. Right, so the first important concept, oops, is not this one, is this here. The first important concept that we need to understand is what it is electromagnetic spectrum. actually electromagnetic spectrum, which is basically all type of radiations that come from a source. Okay, in our case, the main source is the sun. This radiation goes everywhere on what we call waves, and those waves can be classified according to the distance between the peak of these two waves, which we call wavelength, and also the number of waves in the period of time, what we call frequency. Okay, scientists used these informations to classify those all type of radiations in some regions. Here we can see some of those. Starting by the radio is the same one that we normally use for listen radio in our cars, for example. The radiation in that region has very large wavelength. That has this ability to go over mountains, over buildings, and go everywhere. Okay. Then we have the microwave, is a little bit shorter, and is used on the microwave machine in our to warm food, also in our cell phones. Then we have the infrared in the visible region. These two regions we're gonna be talking more later, so I'm gonna just jump it to the next one here. The ultraviolet, and then we have the X-ray. The X-ray is way shorter and has the ability to cross tissue, but not dense tissue like bones. That's why we normally use these in medicine, for example, or to evaluate seeds nowadays. And here gamma ray is very energetic, very short wavelength, and in plant breeding can be used, for example, as one strategy for cause mutation. Okay. Coming back here to visible and infrared region, the majority of sensors that we normally use on plants are on those regions because these regions are very important for physiological process, and we're gonna see some of them, okay? Just for you keep in mind, the visible region comes from around here 400 all the way to 700 nanometers, which is gonna be the wavelength distance, the distance between the two waves that we were just talking about. When this radiation comes for a surface, all those type of reactions can happen, right? So there is the transmission when the radiation just passed the object directly. There is the reflection when have contact with the object and come back. The refraction when change the direction. Diffraction when broke in many other small radiation. Absorption, which is very important, for example, for photosynthesis in plants. And scattering, which is one of explanations of why the sky is blue. Okay, you're gonna agree with me that all those type of reactions happen when the radiation coming from the sun, for example, or from a light, come to a surface, happen everything at the same time. For example, here we have the sun, all this radiation coming, right? And here we have our object. In our case, it is a plant. When you go closer to the surface of this plant, we have here the internal morphological structure in the leaf. And remember the reactions that I just explained. Again, everything's happened here. And are you guys gonna agree with me if we change the internal morphological structure, we also change the way of these reactions happen? For example, the angle of this leaf, or the concentration of guard cells, or anyway, other regions here in the leaf. If we go changing, or more cellulose, anyway, if we change anything here, all those reactions change. So keep this in mind, okay? And whatever's come back, it is what we can collect with sensors. Not necessarily only this reaction of reflectance, but anything that's come back, our sensor has the ability to capture, and this difference is related with the surface from where this radiation is coming back, right? So the fact of coming back, it is what we call reflectance, which basically is the fraction between the amount of radiation that comes to a surface that is reflected back, right? So this is the reflectance. And here in this image, it is on the y-axis represented the y-axis in this graphic here represent the reflectance, which again is the amount of the fraction between the incident radiation compared to what our sensors can capture. All right. On the x-axis here, we do have the wavelength, the distance that we were just discussing about the radiation, right? The electromagnetic spectrum. Right here on the 400, we have the blue, green, 700, the red. And then we have here the infrared, which is the region that we were discussing 1 minute ago. As you can see, and as we saw in the previous slide, depending of the surface or the object that we are looking for, the reflectance is going to change in the spectrum as well. For example, snow has high reflectance here on the blue and visible region, going all the way down in the infrared with low reflectance. Okay. So the radiation that is coming to this surface is not coming back and being captured, for example, for our sensors, which is going to be collecting this information. Again, here we can see the dry soil, right? So the dry soil has this shape. And here on this full line here, we have the vegetation, which has this profile. Just if you are new on these, when you are working with pixels from vegetation in this region, you're supposed to see exactly this profile here, okay? Keep an eye on it, because when I started work on this and I didn't know exactly how supposed to be the shape, I was extracting information from soil, and then my curve was looking more like this other one here. So pay attention to just be aware if you are collecting information from from pixels with leaves. Okay, we saw that there is the source of radiation. This radiation is classified in some regions, right? We use wavelength and frequency to make this classification. When this radiation comes to a surface, there are many reactions. All those reactions, when they reflect back and is captured by our sensors, it is the reflectance, and each object has different reflectance on the spectrum. Okay, we saw the vegetation shape, and also remember, vegetation always have the same shape. However, the amount of reflectance in each region per species also changes, and we can use these, for example, to evaluate the concentration of one species in a forestry, or for example, the amount of land covered with soybean or corn or any other crop, using, for example, hyperspectral information from airplanes or satellites. Okay. In this case, we go, for example, some species has very close reflectance information here, for example, on the blue or on the visible region. However, there are high differences farther on the near. And all this information can be used together, and of course, with validate data to really validate this information and make sure that you are talking about different species. As you can see, with different objects, we can differentiate species with the same principle, but we also can differentiate genotypes in a population, which now we come closer to what we do in plant breeding, right? Because our main idea is try to find, trying to use this technology to identify different individuals in a population and correlate these with our phenotypic information, right? Right. For example, here we can see a wheat population from this paper, and then one genotype has higher reflectance here on the visual on the near region, which means more health, more green, because this region is normally related with greenness, concentration of water. So that means this plant is kind of healthier than this other genotype, which is right here. So when we try to correlate this information, for example, with a disease in the field or for example, maturity or senescence, we can easily compare the data that we collect in the field with the vegetation index or any other UAV trait that we calculate, and try to make meaning of these, right? Try to use this information for the classification. And so this is more close to what we're gonna talk about Bison-Fly in a bit. The same idea that we saw to differentiate objects, species, plants, now we can go even further and try to classify different morphological regions in a plant, right? So here is a multispectral, a hyperspectral image, and this group, these colleagues here, they classify the pixels according to each morphological part in the plant. For example, leaves, here panicle and stalk, and also background. And when they did the same plot, on the x-axis we have the wavelength, and the y-axis the reflectance, we can see that different parts, morphological parts in the plant also reflects different. And we can use those differences in all profile, for example, in machine learning models, and try to classify or each region, each morphological region, and with these developing algorithms or other softwares to get information. For example, once that the algorithm could classify the panicle, it's possible, for example, calculate the height, the angle, the leaf angle, the plant height, and the panicle height. Anyway, many other traits that until now was very hard to calculate or even evaluate for plant breeding. However, with the technology, of course, we need to plan a pipeline from where this data is going to be collected and extracted, and again, give to the breeders for making decision, right? But now it's way easier to account this information for selection, for example, in early stages, when normally we just go for yield or quality, depend of the crop, and okay, those more specified traits we see at the end of the cycle, right? And the end of the breeding cycle in late stages, because it's less genotype to evaluate. Anyway, so this is some important concepts that I want to start before going to the Bison-Fly. There is questions. Feel free. Feelings, feel free to share other ideas as well. Okay? Okay, starting here from our field trials, this is some images and a pipeline that I start working on when I was in Wisconsin, work with potatoes. And we're gonna have here just some ideas about how to prepare the field if you are starting work with UAVs right now and you want to introduce UAVs in your program or research. The first point is what we used to do. I was going to the field. I normally go to the field and taking images from the whole breeding cycle, starting from before the plant starts sprouting all the way to senescence. And then we have here, it's very important if you do not have a RTK GPS connect with your drone, but even I always recommend putting some ground controls just to make sure, right? To make sure that you're gonna have the geographic correction for make sure that your ortho mosaic is gonna overlay during the process. But the ground control points, we basically put them in the field before starting our collecting images and stay there during the whole cycle. Each group do this in a different way. The way that I like to do is keep the same position and the same ground control there during the whole cycle. Then I only need to go there once to collect the latitude, longitude, and altitude for that specific ground control. Here, we were using those leads And this leads is very interesting because they are very cheap and easy to find in the, from in the image. And also it's very close to the ground. And then if a tractor pass on, many times it survives, right? Okay. So as I was saying, we put these in the field and then we go with a high precision GPS and collect this information before start, or actually during the cycle, right? But we need this information during the orthomosaiking step. Also here it is very important have some kind of calibration, reflectance panel for calibration, or some calibration for reflectance. Why this is important? For example, imagine a situation when you have a cloud in your field and the amount of, do you remember about reflectance? It is the amount of, is the fraction that our sensor can capture compared to the incident radiation. When we have this calibration, the softwares know, or the pipeline know, that all pixels right here in this gray square has, for example, 40% of reflectance. Okay, so here's 40% of reflectance, 40% of reflectance, 40% of reflectance compared to the incident radiation that's coming here. In this case, imagine that during your entire flying time, you have this type of light, but then come in a moment we have a cloud, and then some genotype is going to be very dark. However, because we do have this calibration here, and in our drone normally has this light, some drones that has this ability, right? Has the light sensor, we can infer or correct the whole field comparing to be comparable, right? Another application also, for example, when you have different flying days or different locations. So calibrate reflectance panel or reflectance calibration is very important point. I know here there are people that has way more experience with different type. In our case, we were using the one that was provided by the same company that we have our drone. Okay, about sensors, the sensors that I already worked before, and also the pipeline that we're going to show can work as well. It is the RGB, the multispectral, and hyperspectral. So the same pipeline, you can extract information from all those type of sensors, and also thermal, for example, any type of image. Okay. Okay,
sorry, Felipe, I want to just interrupt real quick with a question from the chat.
Sure.
So Max asked, is maintaining the same height and angle of the sensor when capturing an image of the calibrated Reflectix panel important? So is it important to maintain the same height and angle of the sensor?
Yeah, I guess it's very important. It's very hard to have this all the time, but as most as you can keep the pattern of data collection, it is very important. And for example, this image here is the software provided by the company. For example, in this case, when you are capturing this image, there is the simulation of amount of instant radiation that is coming, and then you use these for during the orthomosaiking to correct all the other parts in the field. Yeah, I know that, for example, many drones with RGB only, it's hard to do it. However, it's possible you include in some panels in the field, or like big panels, and trying to do these afterwards during the image processing. Okay, let's continue here. One step that is very important, in our case, we were using field agents to collect data from Centera, but there are many open source softwares as well to do it. But basically here, this is what we call the field map for flying, so our plan for flying. You just select the area where you want to fly, and basically the majority of my flights in potatoes and wheat, I was working with 75% of coverage. For example, 70% of this dot right here is exactly like this one, is exactly like that one. So 75% of coverage that give us also a depth idea during when we are making the cloud, for example, to calculate the digital surface model and have an idea about plant height. We're gonna see about this as well. We were flying 200 feet and 15 miles per hour. So that was the recommendation for when you are flying the entire cycle looking for yield. Okay. And then after we do collect these single images, we make the orthomosaics, which is gonna be something like this. For doing orthomosaics, here there is one open source option, which is the Open Drone Map. And Open Drone Map is a community of an amazing guys, and they scientists, and they share their knowledge with the community about how to do this in an open source way. And for instance, they do have this web ODM, which is a click point interface for the ODM. So if you are more coding guy, you can go directly to ODM, or if you're a more like click point guy for this type of, this type of orthomosaic, you can install the web ODM. It's very cool. And also Feed Imager, which is the R package that I worked before, is part of this community as a suggestion from how to extract information in agriculture, or from orthomosaics actually. Here, just some outputs from this web ODM. You have the orthomosaics, which is normally is the most important, for example, for us now for calculate vegetation indices anyway. And also we have here the DSM, the elevation models that we also use for plant height. They do offer many other outputs here, but we're gonna use mainly the orthomosaic and the DSM. Okay, we're gonna the first part of Bison-Fly is the field imager pipeline. So basically we're gonna use RStudio, and here is gonna be the code, and we're gonna communicate with the software using this plot region when necessary. So the open source suggestion to extract data is gonna be using web ODM for pasting or stick together the individual images collected with the drone until you get your orthomosaic, just following click point steps. And then from this point, you download your orthomosaic and come to R and follow the field imager tutorial, okay, to extract information. Here we go. Basically, what we're gonna do right now is selecting the field that we're gonna work with. We're gonna rotate, remove the soil. Remember that removing the soil here depends on your question. We're gonna talk about this later. And then make the boundaries and extract data from there. Okay? Okay, let's start. We can talk about applications if you have questions at the end. Okay, we're gonna follow Bison-Fly pipeline. The Bison-Fly we developed with the drone to phenom community. Here's Max Fedman, which is part of this project with me, along with Dave LeBauer, Jennifer, Andrew Green, Dr. Andrew Green, the breeder in this spring wheat breeder at North Dakota State University. He was my PI before I'm moving for industry. This tutorial, as you can see, there is the introduction part, and then we have the image analysis, and we start with more statistical analysis to understand how to apply this data, or how to start playing around with this data in your breeding program or in your research. As you can see, All data here is from the spring wheat breeding program at North Dakota State University, and the breeder is Dr. Undergreen. So here is the link for his webpage, okay? For doing this pipeline, you need to install R. In this case, you guys do not need to install because we are using the binder, but feel free to follow the pipeline to install all package that is necessary in our computer afterwards. Here are some fields from where we collect data last year in North Dakota State University. Here's our group and some views from the farm. So when you were starting working with UAV in your programs, the first point is necessary to have a drone pilot certified to collect data, right? For instance, this person needs to get the permission from the FAA. Here's the link. And it is very important to make this exam. Actually, this is the exam that gives you permission for flying and to be a remote pilot for small unmanned aircraft systems, okay? So if you want more information, just click here and start reading what is necessary. Also, if you are working in a university or even in an industry, there is a department that take care of giving you permission to collect information in the farm or anyway, in your field trials. In the case of North Dakota State, we need to fill up this permission and wait for the responsible agree. And so first of all, you need to check in your institution who is the officer responsible to give these permissions and what's the process. Okay. After this, it is very important to have a drone. In our case, in North Dakota, we have this Inspired 2 from DJI connected with the multispectral sensor from Centera called 6X. And connected with that 6X, we do have the reflectance panel that we are just discussing about in our theoretical part, okay? Trying to correct, for example, different days of flying or a cloud problem during the flying time. Anyway, just to make sure that the reflectance that you're receiving there is comparable. After this, we saw that we were using, for making our fly plan, we were using field agents from Centera. However, there is other open source tools, for example, Pix4D Capture and Drone Deploy. So feel free here to come and to check all of them. And normally, if you do not know how to start or which parameters to use for collecting our first images, we do recommend starting with these parameters here: 75% of overlapping, 15 miles per hour, and 200 feet. Again, this is a good parameters when you are extracting average per plot. If you need to see, for example, the tails, counting stand count or diseases, you need way more, for example, you need to fly way more close to your plants and way slower, okay? So just pay attention on this. For do the georeferencing or correct, right? The geographic positioning in our fields, we were using these leads from 5 gallons. In the presentation, we saw the orange leads, and depending on the soil where you are working, the white leads are better to see. So just check before starting putting the leads or the ground controls in your field. Here, just one example, we were using these metal sticks for fixing the lead in the field, and also we were making a hole in the middle to avoid water from the irrigation or the rain. So just for you keep in mind. Here's one strategy that we thought when we have 2 experimental trials right beside each other, and it's hard to identify when start one or where start one and finish other. The main idea here is having on the borders the ground control points, okay? And dividing the field, we can include where is going to be our division point, for example. And in this case, the first row and the first column and the last row and the last column are borders, so we can remove it. You can just put here the lead to for facilitate you during, for example, building the plot shape boundaries or file to click. Okay, we're gonna see this. And here are our RStudio. Okay. Now come the most important point, I guess, in this section is talking about the free data that we have here. Basically, we do have 106 white width lines, right? This is block design. We do have 15 flights data, and this 15 flight data, we're collecting all those days that is highlighting here. And also we do have field agronomical traits, maturity day, plant height, days to heading, lodging, and yield. Okay. As you can see, when you click here and download, we are giving for free, guys. Please feel free to use it. If you do not have, for example, I want to work with UAVs, I want to learn, but I do not have data. That's not an excuse anymore. You have data here for playing around and trying your models or try to improve this pipeline. We do have 15 multispectral orthomosaics, 15 RGBs, 15 DSM files for calculate, for example, plant height. We do offer the CSV file with the agronomical field data. Again, which are the traits in the agronomical field data? Those: maturity, plant height, days to heading, lodging, and yield. And also, we have here the metadata explaining what each mosaic or name of mosaic means, and the environmental or the environmics information from one station very close to Casseton. In our case, our data was our flights were in Casseton, but the closest station to our location was Prosper. That's why we went there and collected hourly basis, I guess. It's every hour we collect from the station main information from the weather, from the soil. So feel free to download these and also start playing around with the statistical analysis. Okay. Anyway, so let's start the tutorial. What we're gonna run is copy and paste from what we have in this tutorial. And then we're gonna see the results together. If you have questions, just stop and make question. All right. Nicole, how many time I have? How long?
You have a little less than 90 minutes remaining.
Okay. Great, so let's start the tutorial. Guys, if you have a question, please feel free to ask. Also, if you have a question about the data, yes, the answer yes, it is free. So feel free to use the data and try to, I don't know, develop a different model, try to improve what we did here and give the feedback to us, and then we can improve the tutorial and help more people, okay? We start the pipeline by extracting data at first. And for instance, we need many different packages for the data extraction and also to calculate the blobs, the blues, and evaluate heritability and principal components. Anyway, we're gonna check this together. We can start, the first part in the code is bring to R all the 15 bands, or for example, the 15 orthomosaics that we have in our folder. In this case, we have, let's check here. we have these 15 flights. How they are classified? They are, I normally I put the number from the sequence of flights. So this is the flight number 1. So before the plant starts sprouting, this was collected at day 4, days after planting 4 in 2021 in Castleton at this day here, 11th of May 11th. Okay, and which type of orthomosaic is these 5 bands, the multispectral 5 bands. The same thing we do have here for RGB, and the same thing we have here for DSM. Okay. This list file has this ability to check in one specific folder all the names, all the files that is inside that. As you can see, we have here all our orthomosaics. The pipeline normally starts by choosing one example, orthomosaic as example, and starting preparing the code based on this, and then you can make a loop to extract all the others mosaics without need to do this one per one. In this case, I choose the image 4, which is this one right here. And as you can see, I choose this in specific because it was when I went to the field and I include there where we're gonna click. Remember those extra leads? I just put this to facilitate during this step, the next step when we're gonna build the plot shape boundaries to know where to click. Okay. One important step is to combine the information that we have from the field. Remember, we have here the genotype names, we have the plot number, and then block, range, and row. We do also are sharing the pedigree. So you have extra information besides having the mosaics, the agronomical information, the environmental information. You also have the pedigree to play around. And here are the agronomical traits. So we wanna combine all this information with the UAV traits that we're going to extract from this image. Okay. How are we going to do this? First of all, we bring the data to R, and then we also have the map. The map is very important because the map gives us an idea about how our field looks like. The map basically it is a matrix with the plot information need to be one's unique ID or unique identifier. For example, I know based on this map that this plot right here, where is my arrow? See this plot right here, it is the plot 1220. Does that make sense? And this plot right below here, this one, it is the 1001. So the map with the unique identifier need to be exactly the position that we are seeing right here. Okay? Based on this, we need also to make sure that our data has one column called plot, for example, with exactly the same identifier. Because what we basically going to do, we're going to do the match. We're going to combine our data with the data that we're going to extract based on the plot identifier. Okay, now how we're going to do this? To facilitate, I'm going to open another plot window. And then for making the plot shape boundary, we do use this field shape function where we give the information which one is our orthomosaic that we are working, right? Our test. The number of columns, in this case, is 11. The number of rows, 20. do we have data from agronomical traits to combine with the data that we're going to extract from this image? Yes, it is the object data. Which column in the data object we're going to use as unique identifier? The column plot. And how we're going to combine this data with our image using the map. Okay, hopefully that makes sense. So when we run this, the function, let me move this here. The function is going to open here the image for clicking, and then we just come here where we put those extra leads, if you have leads or not, right? But make sure to choose exactly the middle of the alley and avoiding the borders. In our case, we have borders. If you do not have borders in your experiment, so just ignore this part. Okay. So once that we did this, the software is gonna make a square, as you can see here, and divide this square or this rectangle, right, by 20 columns and 20 rows. And at the same time, we're combining each one of those plots with the data information that we have right here. Okay. All right. So just for we see now, we do have our Ortho mosaic and our boundaries, well draw in this case. But as you can see, it's a very straight field. Normally breeding programs, we do not see that straight fields, right? In this case, there are some suggestions about what to do if you want to use this method, but you can contact me later and I can explain. It's very simple, but you need to make each pattern region in different, per time, and then you combine those together to make only one shapefile. But I can explain if you have this problem, so just contact me. Okay, the next step now is what we call removing soil. Removing soil here depends on what you are looking for. And we are using here the function called field mask. What I mean about looking for, if you wanted to see any type of relation of cover, for example, leaf area index, which is a proportion between soil exposure and canopy, you should keep the soil there, okay? Because when, for example, you are using a vegetation indexing, in this case, if the inside the plot has more soil pixels, it's gonna push down the greenness in that plot, and in this case, is connected directly with the amount of canopy or the index leaf area index in that specific plot. But if you're looking for only specific the spectrum texture or spectrum performance of each genotype, you should remove the soil from the analysis and only go for the real greenness related with your genotype. Okay? How we are using this function? In this case, there is you can identify 5 layers if you have. Sometimes you do not have, so you do not need to inform anything for these 2. You can inform which trait you wanna use for do the segmentation. In this case, we are gonna use an internal index, but you also can use any other, index that is better for your field. Depending of the soil color, this index is gonna change, okay? And then basically here we are doing a segmentation based on thresholding, where we calculate the index and then we say everything above 0.7 is true, which means more green, right? And everything below remove is false. For example, there is a default, default correction, but in this case, because the soil was way darker than the original one that we used to calculate, you're gonna see that the segmentation is not that great as using any DVI. For example, can you see? All right. So let's keep the original one from the tutorial. Okay.
Felipe, there's another question in the chat.
Okay.
Or two questions, I guess. So the first is, where can someone find information about the order of channels in the image? And is this determined by the sensor or by the program that generated the Ortho Mosaic?
Here we go again. Could you repeat? Where's this question? Where what?
Okay, so this is in chat if you want to read it. It's asking, where can someone find information about the order of the channels in their image?
Mm-hmm. Oh, okay. That's very interesting. That's a very interesting question. And also, this is a very important tip. You should check with the person that developed your sensor, because normally the sensors are classified RGB, red edge, and near. Okay, but some sensors can be classified as blue, green, red edge, and near. Why? Because the blue has the lower wavelength. The best way to check this is in the manual of your camera or your sensor or your drone. If it is a RGB, for example, only the RGB sensor or in your drone, normally the layer 1 is the red and green and blue, okay? But some multispectral can come for the wavelength distance, and then the blue is gonna be the first one. That's a very interesting question. Check the manual. Yeah, that's the best tip. I had this mistake before too. Okay, what the other question?
So the other question is it possible to shrink the plot size after creating the grid in order to remove the alleys and keep only the actual plot?
That's a very nice idea as well. We will work on this right now. The only thing that we normally use to correct for the alleys and shrink, it is a parameter called buffer. Okay, so in this case, the buffer during the data extraction, right? The extract information, the buffer is gonna remove that borders. But we are working. It's just because I do not have time, but I have friends working on reducing the size per seed in the plot, not necessarily only using the buffer. Okay, but for now, concentrating the buffer number here. We're gonna go there right now. Okay.
Okay, and then just one more quick question. So someone got a little bit confused when you were talking about the NDVI. Were you using that to remove soil or plot polygons?
We are using the NDVI to remove soil. Yeah. So in this case, we are using a vegetation indices to identify what is green and what is soil. And yeah, the plot polygons, I'm not sure why. Here. Plot polygon we normally do for only to calculate the plot boundaries, right? I don't know if I answered your question.
Yep, it looks like you answered it. Thanks.
Okay, are you guys understanding, right? Just let me know, I can go slowly. Okay. Well, okay, now, once that we already did this, we already make some decisions here. I did. If you want to have more information about all the steps here, you can check the Open Drone Map, exactly. The field image tutorial. The field image tutorial, we go from how to rotate the image, how to remove the soil, and there are some explanation, okay? How to do the plot boundaries. Anyway, how you can use the original angle, see, boundary, how you calculate vegetation. There are many steps, information that we are not covering here today. I'm gonna put in the chat. If you have a question also, you can contact me. All right. Once that we already did that, we can run all the 14 flights in a loop. Okay, in this case, is a very simple loop where we're gonna tell the software, I'm not gonna run because it takes some time. And this for the workshop today, we're gonna go directly to the final results. But just explaining what we are doing here, first of all, we are calling each Ortho mosaic per time. Each Ortho mosaic per time, we are using the mask to remove the soil, and then we are calculating different indices. Which indices we are calculating here? We are calculating all those indices that is internally already developed in the software, okay? If you wanna any other index that is not there in the software, I mean, is not in this table here, you can use a parameter called myIndex. And in this parameter, you can include any equation or layer combinations for calculate your new index. After this, we're gonna use the information called field info to extract, right? Information per plot. In this case, we're gonna put here our image with all layers, and then the shapefile that we just draw. And again, answering that question, previously, we have this parameter called buffer, where in this case, because our image, it is in meters, we are removing 5 centimeters from each boundary, okay? 5 centimeters for each side here. And then we try to avoid, for example, regions where there is overlayer, I mean, leaves overlaying each other. And then we use this function called field area. Field area is going to calculate the canopy per plot. And again, we come here with the plant height. How we calculate the plant height? We calculate plant height this way, right here. Remember that we have 15 flights. The first flight is before the plant starts sprouting. In our case, we collect with 4 days after planting. That's gonna be our time 0 and our reference height, soil height. Then we're gonna come back there, and when we have leaves, and collect exactly this the same way, right, with the digital surface model. And as you can see, we do have this higher region here, tallest region in the soil. This region is more green, and here we have the same higher region. I don't know if I can say this way in our trial. If we extract data directly from this image, that we're going to see that all those genotypes in that specific region is taller than the genotypes in this other part in the field. But then we use here what we call canopy height model, and we remove from the image with plants, we remove the base, and what we have here, it is the height connected with the plant. As you can see, pixels were supposed to be soil come all the way to around 0. Before was the original altitude, okay? Because we have here on the x-axis latitude, on the y-axis longitude, and each pixel value it is the altitude. And when we remove one from other, what we have, it is the canopy height model, which is more related with the original height. Then it's possible to remove the soil using the mask that we calculate using the RGB. And basically it is what we are doing here. And then extract the average or the quantile per plot. Does that make sense? It's exactly what we did here. So we have the time 0. See, it is the only object that is fixed on the image 1, because image 1 is our soil base. And then all the other 14 flights go changing in the code, but it's all comparable or being related with the first Ortho mosaic. Then we do the canopy height model, removing the soil base from the original image that we just saw. Again, we remove the soil using the mask that we calculate with the RGB image. And here we have the new mosaic, the new the new canopy height model image, which is going to be this one. With this, what we can do, we can use the function field info to calculate the quantiles. The quantile is going to give us 0, which is the minimum value, pixel value in the plot, 25, 50, 75, and 100. The 100 means the maximum pixel value in the plot. However, in the literature, is normally we see also not on 75, but also we saw at 10% and 90% of the quantile. Here's the code also for you guys do this, okay? And then basically we just combine everything in one table. Just change some names. Anyway, at the end, what we have, it is this table here, what I'm calling data total. How this is classified, we have the name from the picture. So this is for example, the flight 10 that was collected at 52 days after planting. Again, casted on 21 and in this day, okay? We have the plot name. This is our data that we combined originally with our shapefile. Plot name, range, row, pedigree, our agronomical traits, the 5 agronomical traits. And again, and now we have all the UAV traits that we extract per plot, right? So we do have here the blue, the 5 original bands, and then all the vegetation indices that we extracted. We have the canopy, remember field area function. And then we have the height. Height 0 means minimum pixel value in the plot. 55 and go all the way to 100, which is the maximum value, and the extra 2 heights at 10% of the quantile and 90% of the quantile, very common papers. And of course, here the day after planting, 52. Okay, if you go down, we're gonna see all the flighting days after planting in only one table. And then with this, you can play around and do some stats. Okay, so I went kind of fast in this loop because there is other videos online where I explain how to do it. You also can contact me and I can send the link to you guys, okay? For example, you can find this tutorial in the Phenom4 YouTube channel. If you come here to Phenom4 YouTube channel, in videos, the first video that we made 1 year ago is gonna be this tutorial. or not this tutorial exactly, it is another data set, but I'm gonna, in this video, I explain how to do the loop and what each one of those steps means, okay? I'm gonna send here in the chat as well. There is a question, why do we need plant height at 10 and 9% of the quantile? Actually, that depends of your question, you know. Normally in papers, we say 75 and 90% of the quantile, because they are more realistic with the reality. For example, when we have the height, for example, the cloud height, the pixels height value there, which is gonna be our altitude, but without the soil. In general, the canopy, where is 90% of the canopy is covered, that's our average plot, average height in the plot, you know. You can see afterwards, for example, when you compare with the visual data that you collect in the field, that you have higher correlation with 90 and 75, more than the average. Okay, because the average you are accounting, for example, also when the leaves go down on the borders in the plot and starting connected with the neighbors. So when you take the 90s, you are taking more the information up high. And 100%, maybe if you are working with corn, you're taking pixel from the tassel, which is the maximum height there, which is not true as well. So 90, 75, 90 is more close to the reality. That's why. Does that make sense? Hopefully, yes.
Okay, Felipe, there's one more question in there. Oh, and Smitha replied that yes, so that makes sense. Okay, so can you describe about the generalization of the proposed method for other fields and projects?
Generalization from other fields and plots? You mean the loop?
I'm not.
Yeah,
I'm not sure.
The generalization is for different crops or for the other images from the same field? Because if it is for the same field, we have the same soil colors. In this case, the way that we are removing the soil there is gonna work just fine because we have the same soil color. When you go for a different region or field trial where the color changes, the soil color changes, probably you need to adjust the way to do the image segmentation, right? To remove soil, and maybe you're gonna change the index or change the value there. That's one point, okay? And the other step is just a generalization because we basically have the steps there, and the steps is all, in our case, automatic inside. There is not much segmentation to do. Yeah, did I answer your question? Feel free to ask again. Okay. Let's continue here. Nicole, do you guys normally make a break, like 2 minutes or something like that?
You know, it's up to you. Sometimes we do, sometimes we don't. It's just kind of whatever you're feeling like. We are recording this. I put this in chat as well. So if anyone does want to go back and view the recording at some point, it'll be on the same page that you registered on.
Okay, perfect. So let's continue here. Okay, we saw the data extraction part. Okay, hopefully you guys understood. If you have questions, you can send here or contact me later in social media. I'm trying to answer everyone in LinkedIn. Now we're going to start more a simple stats or statistical analysis after you extract your information. And that really helps, for example, when you have no idea how to start. Here we're going to start our statistical analysis using evaluating our field data, agronomical data. Again, as you check here, this is our table, right? And we do have these agronomical traits, which normally, which are the main traits that breeders use for doing selection. What we first need to see is how important it is each one of those traits, and also what the heritability of that trait, those traits, because that also helps if, for example, is a high heritable trait or not, how expensive is to collect that trait, and how, for example, changing the ways of work for collecting that specific trait could improve genetic gains. Okay. What we're here's just a loop again for calculating everything at once, just to explain what we are doing here in a very fast way. So take your time to digest and go slowly in the code here. But we are using the most simple model, which is going to be basically the trait compared to the genotype, and like in complete design. And here you can also play around and check with the including some range and row structure. You can play around and put the range and row as random. Anyway, feel free to check. Okay, so for strategy, we decide just keep the most simple model. Okay, what we are using the software LM4 and the function LMR, which allow us to do mixed models. Once that we, and then here basically we are extracting we are calculating the heritabilities and to check how each traits in this population, how the heritability for each trait in this population. And then again, we now are using the genotypes as fixed effect and extracting the blues, and those blues are actually the adjusted means we're going to use for doing the second analysis, like principal components and correlations afterwards. Okay. So the first step before we starting comparing, or actually compare with the UAVs, we check the heritability using this simple model. Of course, remember, this is just example. For our traits, we can see that days for heading is very heritable, very high heritable, and yield normally is very low heritable, as you can see here. The other ones is on average or moderate heritable. The same thing that we did for the agronomical traits, we're going to do now for the UAV traits that we just calculate. In this case, we are calculating the heritability per day of flying for all those traits that we collect. For the single bands, some people like to work with the single bands, besides do not have a directly biological meaning. Okay, we have the we're gonna calculate the heritability for all vegetation indices that has a biological meaning, because scientists went there, check how the plant perform in the spectrum, and combine this information. They come up with formulas or equation that has a biological meaning, which is related with chlorophyll content. Anyway, many different explanations there. We're going to calculate the heritability for canopy and all type of plant heights. Okay. And for instance, you can see that we do have here a code for plotting this together. And you can check in a bit. What's very interesting, guys, if you have time to come in this code and change the model and try to make these improve, try to increase the heritability, that's very nice, and is a very great feedback, for example, where we can include the model that you suggest in the tutorial, and this is extra option for if there is anyone there that want to learn. Because we do have what we have here. We have Okay, what do we have here? On the x-axis, we have the days after planting, okay, starting from 4 days after planting all the way to 80 something. In the y-axis, we have the heritability value, okay? Each box, it is one UAV trait that we calculate. Right, so what do we have? As you can see, there is this tendency of stability in the vegetation indices. Some days here were lower heritability. What I want to get your attention is, look, if we go only with the heights at zero quantile, which is going to be the minimum value in the plot, we almost can't explain anything, right? So the maximum pixel height value there is more, heritable, or, is easier to explain. But check this. Something happened at 70 days after planting, 73 days after planting, that brought our values all the way to 0. Okay, so keep an eye on this, keep this in mind that we're going to talk about this later. All right. Okay. Continue, once that we check the heritability, continue here in our tutorial, we do discuss, let's come back there. We did discuss about, see, all the steps that we just saw, okay? Heritabilities. We did discuss about the importance of area under the curve. The area under the curve, it is a trait that we normally calculate when the whole cycle matters. For example, if you are looking for maturity, it's basically only the flights at the end of the cycle that is more related with senescence or maturity, right? Vine maturity. However, if you stop to think on yield, the information during the whole cycle is more connected with the plant performance than only one specific flight during, or a specific moment during the season. Our question was, how important would be combine all flights in only one information, and then besides making selection or using 15 flights, for example, on my model, I can use only one information. The way to calculate this, we go for the area under the curve as a suggestion, right? Let's run the code here. But in this case, I just choose like 6 flights to not use all flights because we do not have space in the plot area. And then we can see that each flight per se follows some distribution. Of course, at the beginning and at the end of cycle, we have more brownish or darker pixels. That's why, because this is a vegetation index, that's why this is pushing back towards 0. And here around 50, 70 days after planting, this moment is more during the flowering time. The plant is very green. That's why it has higher, vegetation index or NDVI values. What we are talking, basically, it is what we describe here. Imagine a situation where, for example, this genotype, the Glen, because has during the whole cycle is less green, for example, than the NED Pro or Fowler, which is has more green during the whole cycle. Imagine if this information of this area below the curve is related with the yield in the end. Our question was that. So, and then we decided go for those traits with the area under the curve. Okay. One important point is checking the heritability from all the flights, the image that we have before, and see which traits or which flights you're gonna bring for this this moment for this area under the curve. For example, flights with low heritable moments or low heritability, maybe you should remove. Okay, there is could be some mistake over there. Does that make sense? Okay. Well, now we're going to calculate the area under the curve. In the package, there is this function called field AUC, which is going to use the output from our tutorial, right? Extracting data there. And it's going to combine all the days in one folder. Okay. And then, of course, we calculate again the heritabilities and check. Actually, here we are seeing how the data distribution from the area under the curve. And then we can, again, calculate the heritabilities and check how heritable are those traits. They are under the curve. For example, it's already more heritable than the yield. As you remember, that is around 30%, and here 6%. Now we need to check if that has any connection with the yield, right? Okay, any questions about area under the curve? We have a better explanation here in the tutorial, the Bison-Fly tutorial. Okay, talking about this. All right, principal component analysis. Principal component analysis always gives us a good idea about how we can see our population in multivariate spectrum. In our case, the way that we were thinking was choosing some UAV traits and also some agronomical traits and check how those are related in the multivariate spectrum. For instance, we use this function call from the package factor extra, and the only change, we classified our check varieties in black and our individuals in the population in gray. And what's happened, for example, max is our maturity reference. That's why, for example, late maturity reference, that's why take more time for maturing. As you can see, it's way down here. And the other side, some of those genotypes are very tall and they can have higher lodging. See how lodging and height is kind of connected somehow. Yield and the original height, it is also collected, is also very high heritable. So basically this map, for the ones that in a very simple way, the way to have an interpretation of these, like acute angle means positive correlation, okay? 90-degree angle means not correlated, and obtuse or all, different directions. Obtuse angle, I don't know if this is in English. Anyway, it is negative correlated when we are only checking the dimension 1 and 2 in this case. All right. And it's very interesting to see that we do have in this population high heritable traits, or high yielding genotypes with a lot of canopy. I do like canopy a lot. It's always well related with yield and height, for example. And when you combine canopy and one vegetation indices, you have a very great idea about yield as well. Okay, this is more a visualization step in the process. Do we have questions? Think.
Yeah, I'm not seeing any in the chat. No, not at this time.
Okay, hopefully you guys are understanding. Okay, now we're going to talk about correlation. Okay, the correlation, there's some code here to prepare the data for to calculate the correlation. In this case, I'm suggesting to use the package Agricoli, and there is this function called correlation. Again, this is just one way to do it, not necessarily the best way to calculate correlation, but it is one start. And to have a visualization of our data, we are using the package core plot. This function core r plot. So let's check. Oh, this is the I guess I have better in the tutorial. Let's see. Okay, no. Let's check the results here in the check the results here. Okay. This is the correlation graphic at 73 days after planting. What I want to get your attention here is, let's check lodging information, okay, with plant height. Wasn't correlated, right? So if it's not here, means that it's not correlated. So the lodging information there and height wasn't connected at 73 days. Canopy and yield was 58 correlated. Look at plant height and the other plant height and the 9% 45 correlated at this specific day. Maturity at 73 days. And for example, canopy and this index, Siri, oops, I'm checking wrong here, is 89. See, is very correlated maturity with these vegetation indices at 73 days. Okay, so let's check now at 74 days after planting. You guys remember that 74 days after planting, 70 days after planting our heritability, our plant height heritability go all the way down in the previous map. Remember that lodging wasn't correlated at 75 days, and now we have like negative correlation with height around 45. That means that the plants, the tallest plants that we calculate with the index were have the lower value for lodging because they were negative correlated. Okay. Just keep this in mind for when we start to talk about lodging in a moment. And this is the correlation using the AUC, so the information during the whole cycle. And for example, when we use the information the whole cycle to evaluate height, we increase to 65% of correlation in this specific case. And yield also increase when we compare with canopy and, for example, this vegetation index. Let's check, for example, maturity day, 72 days for shading, 62. Anyway, just for have your time to go slowly in each one of those steps, okay? Do we have questions here? We do have one question. The significance is also shown in the correlation matrix. Yes, yes, this is the significance. Yeah, so blue means positive and red means negative, and the intensity of color also means so how the value, how correlated it is. All right. We check here, all these are really preliminary evaluations before we really go deep on, for example, applying these, using these to apply selection, okay? Those are what we call preliminary evaluation to check the data. Again, if you change the model there, you're gonna change those values. Feel free to play around and see how this go changing. Now we're gonna talk about lodging. So what's happened? Here's how how people in wheat do lodging classification. But what's happened actually was at 73 days after planting, I went to the field and I flew. And in that night in North Dakota, we had a really strong storm. And at the next day, at 74 days, when I went there, look how many genotypes in our field looks like. Many of those suffered lodging that day, right? So we flew again at 74 days. And then we use this data to do some evaluation. For example, that's a couple days before, and then that was at the 74 days when we flew again. So how we can use this information to evaluate lodging. All right. In this case, we are selecting here 1 day before and 1 day after the lodging, and 80 days to check how the plants behaves afterwards if they have the ability to come back, for example. Let's check here. Basically, we are doing here some code for preparing the data and combine the data in a way that we can make graphics. And we come up with this graphic right here. So as you can see, we just make a box plot with the original data, and we do have here one check variety, which normally is a taller plant. Oh, on the x-axis, actually, let's start from this. On the x-axis, we do have the lodging scores from 0 to 3, and the y-axis we have the estimated plant height from that specific day at 9% of the quantile. We do have here our reference for height and not lodging. This is a genotype that has this is never lodging, is a taller plant. And according to, we increase the height that we calculate with the drones, see, increase the height, we also increase the lodging score. For example, at 73 days, the tallest average genotypes, they receive score 3. However, in this night, we have that really strong storm, right? And then many genotypes lay down. Many of those were the genotypes that received score 3. And when we flew the drone again and calculate the height at 90% of the quantile, check what's happened. The genotypes that were receiving score 3, or I mean, they normally lodging, we could capture this height reduction average in the plot. So they were shorter than the other genotypes that tends to stand, continue standing. And then we flew again at 80 days after planting, and we checked that those genotypes that actually lodging, the average, many of them tend to come back to their straight position, and we could capture this without need to go to the field directly and collect that information again. So here we have two important informations. We can identify this lodging effect. See, the average comes from the tallest all the way to the smallest group, the shorter group. And also we could capture the ability of some genotypes to come back to the straight position. There wasn't a trait easier to collect before, but now you can evaluate these, and also, for example, starting combine these with molecular data and try to find genomic regions related with this ability to come back. Okay, here just some suggestions, okay, guys? All right, let's talk about maturity. In the tutorial, there are more steps there and other code to play around as well. So maturity, we normally go to the field and classify the genotypes as early maturity when 50%, we explain here. When 50% of the plants has this peduncle yellow, right? So if it's green, like here, is not late maturity, for example, and if it is yellow, 50%, it is already mature. What we basically did here, we select flights around the maturity time, and we check how the data is connected, maturity data is connected with two vegetation indices that evaluate greenness, right? The amount of green in our in the plants. Again, we just prepare the data. This is just some code. But what matters here is to check the regression, the simple regression. As you can see, at this point, there is not much correlation or, linear regression between the index that we are calculating, which is this 2 index, and the days to maturity. However, according to the plants really starting getting more yellowish, we starting seeing more visualize this linear regression and starting find, for example, some meaning of using vegetation indices to do a more quantitative classification for maturity in an easy way. Because normally here you go to the field many times during the season. I mean, every 2 days you collect data from all your genotypes, right? That's a very interesting way to do maturity and saving time as well. So it's a point that is also well developed in potatoes. In this case, in potatoes, we're a little bit easier because there we do not go for days for planting. We go for 1 to 9 at one specific day, right? So after we give this score to maturity at around 100 days there, around 100 days, 120 days, we kill the vine. For example, we go in the field and classify 1 for genotypes that is already dead, all the way to 9 when genotypes are still green. And then here is more a categorical trait, and we change this for an index, right? Which is more quantitative. And we have very nice results. And also we could capture using GWAS the same genomic region than the vine maturity given visually, right? Because what's green to you is not maybe green to me. And we always include some bias in doing manual visual score, but the drone or the image actually not, is more, we keep a pattern. Okay. Okay, do we have questions? Okay, let's continue. You can stop anytime. The last part of our conversation actually is trying to use this information to UAVs in a different way to understand yield. And in this part, we are calling back here all the data, right, that we calculate. And we have, we are seeing the QQ plot from the residuals to check if our model is really explaining the variability that is over there, right? So if our residuals follow what is expected. And the main idea now is to include one of those vegetation indices or UAV traits in the same model that we calculate yield. For example, we are including here one trait as fixed effect, right? Each one of those traits is as a fixed effect, just as a covariate in the model. And the idea was to check if we could reduce the AUC in our model. So the AUC give us an explanation how well the model is adjusted for the data that we have. And as you can see, using, for example, at one flight at 55 days after planting, or the area under the curve, we could reduce the AUC, or which means like lower value is the AUC better, your model is adjusted. So we could reduce the AUC, AUC, no, sorry guys, AIC. Am I pronouncing this right? AIC. Okay. We could reduce the AIC, and meaning that including any one of those traits in your model, when you are modeling yield, for example, you are fitting better your data. Does that make sense? So only the fact that you are including one column as a covariate in your model with this extra information, you're already correcting better, fitting better your data and have better results to evaluate yield, to make selection based on the yield that you are, for example, based on blue or blob that you're extracting there. Okay, this is the first point. And the second suggestion here is what the possibility of you doing indirect selection using one of those traits. Again, we use here a very simple model. It's very important to include, for example, blocks effects, rural range. I mean, changing and increase, do the phenotypic mixed model a little bit more complex before coming to this step. But again, we have here our yield only compared to genotypes, right? We are extracting information, and then we're gonna compare what's happened if I apply selection using the area under the curve information for all those traits directly. When I compare the rank, when I rank the yield, and when I rank those traits, am I selecting exactly or the same genotypes? What the proportion of the genotypes that is being selected similar? And what we have here, we are only selecting 25% of, actually, no, let me see. So basically, when we do the rank of genotypes in yield and those traits, for example, for this index, CRA, we are selecting 61% of genotypes in common. That seems very low. However, we were selecting a very short group, right? So this is a very short population, 100 genotypes, and not necessarily 100 genotypes, right? So selecting 20%, or actually 10% of that's gonna only 10 genotypes, that really can reduce this number. However, when you go for early stages in breeding, when you have 1,000 genotypes or over 1,000 genotypes, what is important to keep in mind is what is good, continue good, when is bad, continue bad. Maybe we are not applying selection to select the best, but we can apply selection to remove the worst, okay? When you have short or smaller populations like the one that we are using here in the example, normally those values are of indirect selection in common is lower. However, what we want to do with this tutorial is give an idea about how to use this information, okay? Is a start point. And basically it's exactly the same, however, now using the area under the curve information. The first one was using the flight at 55% of 55 days after planting, and now is using the area under the curve. Almost didn't change. So the same genotypes were continue, greenest or have the higher canopy during the whole cycle. We do have a better explanation about this step in the tutorial when you come to this part right here, like using UAV data for doing to evaluate yield and applying selection. We do explain here the AIC, right here, and explain how this fits better when you only want to use yield, bloop, or adjusted means for applying selection. The fact of reducing this value means that your model is better fit. And just some idea about indirect selection. And unfortunately, this is a small data set, and we couldn't see a very strong value that is more, sellable. It's easier to sell the idea. But anyway, try to do these in your data and check how that increase. Also change the model to see, change the model originally, right? To see how this can increase as well. All right, so that was what I have planned for today. Here we do have a forum for questions. If you have questions, suggestions, how to increase it, please feel free to contact us and give your suggestion. Everything here is free, open for using. Our main idea is trying to develop a tutorial where people can at least start and go improving from that point. And feel free to also help us to improve this tool. Thank you here, David Lebauer, Jennifer, Max, for your support. Thank you, Dr. Green, for giving to the community this huge data set and so complete information. Thank you also, AG2PI, for the seed grant that allow us to develop this work as well. And what else? If you have more questions, feel free to contact me on LinkedIn. I'm always posting all information that I know there and trying to help the community the way that I can. I'm always answering questions as well. Feel free to contact me. If you have more information about, if you want more information about phenomics, I also recommend following Phenomiforce and checking all tutorials that is there. For drones, we have the entire season, like over 10 videos with drone image analysis in a different way. All right, Nicole.
All right, thank Matias, or thank you, Filipe. Yeah, does anyone have questions? Feel free to unmute yourself and go ahead and ask. If you'd rather do that, then put it in chat. Otherwise, thanks for attending and your participation in AG2PI.
Yeah, thank you guys. Feel free again to give feedback. Feedback is very important for we improve this tutorial for the community. This is not for only for North Dakota there, but it is a start point for everyone. And if you think that we can include an extra step in the tutorial there or a web page that may help someone else, just let me know. We can also change the code or change the the description. Yeah, hope you guys enjoy it and learn it. And I feel glad to answer any question afterwards.
And we do have a survey. Once you leave Zoom, it should pop up on your screen. So please do fill that out. And that information will be shared with Filipe as well.